Okay, we're on lesson 3, DNA and genetics. Let's look first. Let me just actually do a little bit of, there we go, see it better now. All right, so first we're going to look at our reading guide and our essential questions. So what is DNA? And that is something that I think you watched a video on yesterday. I'm trying to think back to my lessons. If not, maybe today. Okay, so what is the role of RNA in protein production? So this RNA is something, we've seen DNA, we haven't really talked about RNA. How do changes in the sequence of DNA affect traits? And so we're going to be talking about what the sequence of nucleotides in DNA does, like what is it coding for, and so then how does that affect traits? It's all in this lesson. So here are some important vocabulary words. DNA, which we've talked about, is deoxyribonucleic acid, nucleotides, replication, RNA, which if DNA is deoxyribonucleic acid, RNA is ribonucleic acid. So the deoxy just means that it's missing an oxygen. It looks like RNA, but it's missing an oxygen from the ribose. Okay, let's see, transcription, translation, and mutation. And these are some big concepts. We're going to spend probably about a week and a half or 2 weeks on just this lesson, because I really want you to get this. I want you to be ready for biology next year, and these are some big concepts for you. All right, so what is this strand? What color are your eyes? How tall are you? Traits are controlled by genes, but genes never leave the nucleus of the cell. How does a gene control a trait? Well, this strand and others like it hold the answer to that question. So we'll be finding out what that strand is. Okay. We're not doing the launch lab, so we're going to just skip that. Let's look at the structure of DNA first. So have you ever put together a toy or a game for a child? If so, it probably came with directions. I know some of you are probably, like my sons were really into Legos when they were younger. So maybe some of you followed the directions to put together Lego sets. And so that's what they're talking about. So cells put molecules together in much the same way as you might assemble a toy. They follow a set of directions. Genes provide directions for a cell to assemble molecules that express traits such as eye color or seed shape. Recall from lesson 2 that a gene is a section of a chromosome. Chromosomes are made of proteins and deoxyribonucleic acid, or DNA, an organism's genetic material. A gene is a segment, so a part, of DNA on a chromosome. All right, so if you look at this chromosome here, it's a bunch of coiled DNA, and you can see the helix here, the double helix down here. All right, so cells and organisms contain millions of different molecules. Countless numbers of directions are needed to make all those molecules. How do all these directions fit on a few chromosomes? The information or directions needed for an organism to grow, maintain itself, and reproduce is contained in DNA. As shown in Figure 12, strands of DNA in a chromosome are tightly coiled like a telephone cord. I don't know if you've ever seen a telephone cord before, because that's kind of before your time, but when I was a little girl and we talked on the telephone, it actually had a cord that was coiled. Hopefully you've seen that. Anyway, or a coiled spring. This coiling allows more genes to fit in a small space. So key concept check, what is DNA? So our answer to what is DNA, of course, is deoxyribonucleic acid, but it's the organism's genetic material, and it provides information that's needed for the organism to grow, to maintain itself, and to reproduce. All right, let's go up here and talk about the molecule that is DNA. Let me see if I can a little bit closer. All right. And what's the best way to fold clothes so they will fit into a drawer or a suitcase? Scientists asked a similar question about DNA. What is the shape of the DNA molecule, and how does it fit into a chromosome? The work of several scientists revealed that DNA is like a twisted zipper. This twisted zipper shape is called a double helix. A model of DNA's double helix structure is shown in figure 13, and we'll look at it down here in just a moment. How did scientists make this discovery? Well, Rosalind Franklin and Maurice Wilkins were two scientists in London who used X-rays to study DNA. Some of the X-ray data indicated that DNA has a helix shape. American scientist James Watson visited Franklin and Wilkins and saw one of the DNA X-rays. Watson realized that the X-ray gave valuable clues about DNA's structure. Watson worked with an English scientist, Francis Crick, to build a model of DNA. Watson and Crick based their work on information from Franklin and Wilkins X-rays. They also used chemical information about DNA discovered by another scientist, Erwin Chargaff. After several tries, Watson and Crick built a model that showed how the smaller molecules of DNA bond together and form a double helix. Four nucleotides shape DNA. DNA's twisted zipper shape is because of molecules called nucleotides. A nucleotide is a molecule made of a nitrogen base, a sugar, and a phosphate group. Sugar-phosphate groups form the sides of the DNA zipper. The nitrogen bases bond and form the teeth of the zipper. As shown in figure 13, there are four nitrogen bases: adenine, which we use an A to represent, cytosine, or C, thymine, or T, and guanine, or G. A and T always bond together, and C and G always bond together. Our reading check says, what is a nucleotide? Well, a nucleotide is the molecule made of a nitrogen base, a sugar, and a phosphate group. And so the nucleotides, let's look down here. A nucleotide is composed of the sugar, which we said was either, when we're looking at DNA, it's deoxyribose. Ribose is the sugar. Or if we're looking at RNA, the sugar is RNA, there would be ribose, just a ribose sugar. Okay, so we've got the sugar and the phosphate that are connected together, and on the other side of the sugar is where the base is connected. Okay, the nitrogen base. And so again, there are four different types of nitrogen base in DNA. And we'll see in RNA, three of those are the same, but the thymine is different. It becomes uracil. It's just a little bit of difference between DNA and RNA. So let's look at the other parts of this. So it says nitrogen bases form the teeth of the helix. So if you think about it as a ladder, these are like the rungs of the ladder as you step up a ladder, and these are the sides. Okay, so the sides are formed from the sugar and phosphates, and see how this phosphate, the phosphate from this one nucleotide, think about this as the nucleotide, is connected to the sugar of the next nucleotide. Okay, and then the same thing up here. So this sugar is connected to the next phosphate. And see how they go opposite on the other side? So here on this side, it's starting with phosphate, going up to sugar. You could think of sugar to phosphate. And it's going opposite on this side. Okay, so one side of the DNA is facing one direction, and the other side of the DNA is facing the other direction. Okay, on page 428, we have a DNA strand here, and let's just kind of look at the picture in figure 14. So before a cell divides, its DNA is replicated. So to replicate something just means to make another thing just like it, right? So we talked about DNA replication and how that has to happen in the cell before it undergoes mitosis or meiosis. So this is what they're talking about when we talked about how the chromosome starts to unravel and it gets really spaghetti looking, and it's called chromatin. That's what's happening during this replication, right? This is the part that we're going to talk about now, is what's happening during that during that part of the cell life where it's getting ready to divide. So this is something that the cell has to do anytime before it divides. All right, so it says DNA strands separate, so it's coming apart here, and nitrogen bases are exposed. So now there was a hydrogen bond that was connecting these different bases, the thymine and the adenine, the guanine and the cytosine. So there were some hydrogen bonds going on here, and those hydrogen bonds get broken, and there's a molecule that does that, but we're not going to get into it. You'll learn about it next year in biology. But number 2, nucleotides move into place and form new nitrogen base pairs. Okay, so these nucleotides, again, was composed of the base, the sugar, the deoxyribose, and the phosphate, right? So this is that group of 3 things. That whole nucleotide is coming in, and so the sugar and the phosphate, the sugar from one nucleotide connects with another phosphate from another nucleotide, and then the 2 bases that are coordinate pair, or they're not coordinates, but they're pairs of each other. So like the guanine is going to connect with the cytosine, and the thymine is going to connect with the adenine. So the ones that are paired together will come in and connect, and their sugar and phosphates will connect as well to form a full new new DNA strand on this side, and then it's going, the same thing's going on over here. And so as they separate, this side and this side become 2 new strands. So half of this strand was the old strand, and half of it is going to be the strand that's being put together, if you will. Half of this strand was the old strand, and then half of it is the strand that's getting put together as the DNA is picking up these nucleotides from just from the nucleus where they've been made and placed. Okay, all right, let's see what your book says about how DNA replicates. So cells contain DNA in chromosomes. So every time a cell divides, all chromosomes must be copied for the new cell. The new DNA is identical to existing DNA. The process of copying a DNA molecule to make another DNA molecule is called replication. You can follow the steps of DNA replication in Figure 14. First, the strands separate in many places, exposing individual bases. Then nucleotides are added to each exposed base. This produces 2 identical strands of DNA. So what is replication? Replication is the process of copying a DNA molecule to make another DNA molecule. Okay, so we just talked about how DNA replicates itself. So one thing that DNA is used for is for passing on genetic information from one cell to the next, or from one organism to the next. But the other thing that we need that stuff for, that DNA for, is to make proteins. And so the process of making proteins, I kind of want you to think about it like DNA is a recipe book. And when we open it up and we can read it, then we can write out a recipe. Like if you go over to your grandma's and you want to get a recipe from her, she's not going to give you her whole recipe book, but she'll let you open it up and read it. And so this process of opening up and reading it and then writing out a recipe on a card is going to be something called transcription. And I want you to think about that word because it has the root word script in it. Okay, this root word script means to write. And so if we think about this DNA, if we think about this DNA as a recipe book, and then the recipe that you write out at your grandma's, that's going to be like a type of messenger RNA that sends information from the nucleus out into the cell, and that information or that recipe is then used by the ribosomes to make protein. So that's what we're looking at now, is what is happening to create the written recipe. And then we'll look at something called translation, which is really just like making the recipe, like taking that recipe home to your kitchen and using it to make a dish. All right, so let's look at making proteins. So recall that proteins are important for every cellular process. The DNA of each cell carries a complete set of genes that provides instructions for making all the proteins a cell requires. Most genes contain instructions for making proteins. Some genes contain instructions for when and how quickly proteins are made. Junk DNA. Well, as you have learned, all genes are segments of DNA on a chromosome. However, you might be surprised to learn that most of your DNA is not part of any gene. Remember, a gene is a segment that's coding for a particular protein, and that protein is going to establish a trait in an organism. So, for example, about 97% of the DNA on human chromosomes does not form genes. Segments of DNA that are not parts of genes are often called junk DNA. It is not yet known whether junk DNA segments have functions that are important to cells. So that's something interesting, I think. Maybe one day you guys will become geneticists and learn more about junk DNA and the role they play. All right, so the role of RNA. Okay, RNA, remember, it's a complete ribose. It's got a complete ribose sugar in it. So that's why the R is different from the D. All right, so the role of RNA in making proteins. How does a cell use the instructions in a gene to make proteins? So remember, we're talking about how do they use that recipe that's inside of the book, right? Grandma's recipe book. How are they going to use it? So proteins are made with the help of ribonucleic acid. It's a type of nucleic acid that carries the code for making proteins from the nucleus to the cytoplasm. So RNA also carries amino acids around inside a cell and forms a part of ribosomes. All right, so RNA has a lot of different jobs, but the part that we're going to talk about, the movement, go back here, that carries the code, that's a messenger RNA. And so the carrying the code is the messenger. The type of that carries the code for making proteins. RNA also carries amino acids around. Okay, the type of RNA that carries the amino acids are called transfer RNA. Okay, and then they also form part of the ribosomes. So let's look at this. So RNA, like DNA, is made of nucleotides. However, there are key differences between DNA and RNA. DNA is double-stranded. Okay, so it's got two of these. Two of these strands, right? There's one and two. So RNA is not. It's just going to be a single strand. So just one side of that double strand. All right, so that's one big difference there. Let's see, DNA is double-stranded, RNA is single. RNA has the nitrogen base uracil, and I talked about that just a moment ago. So instead of the T, the thymine, it has something that we call U for uracil. And the sugar ribose instead of deoxyribose. Again, that deoxy just means that it's missing an oxygen. But RNA has a complete ribose sugar in it. All right, so the first step in making protein is to make so messenger RNA from DNA. The process of making this messenger RNA from DNA is called transcription. Remember, that's writing out the recipe from Grandma's recipe book. Let's see, figure 15 shows how messenger RNA is transcribed. Transcribed is just the word that means going through transcription from DNA. So what is our key concept check says, what is the role of RNA in protein production? Okay, so the role of RNA in protein production. First of all, it acts as a transporter of the recipe, right? It's a messenger. It takes the code, or it carries the code for making proteins from the DNA in the nucleus to the cytoplasm. So it's kind of like that recipe that you wrote out. Then out in the cytoplasm, you have transfer RNA that carries around amino acids to the ribosomes, and then you have ribosomal RNA that that puts those amino acids together into long strips that eventually become proteins.